Hi, everyone. Welcome. As you may already know, we have two seminar series that running around 4 p.m. on Mondays in parallel. One in physics colloquium, and the other one is structural quantitative biology seminars in the QB3 building. And once per semester, we have joint seminars between physics and SQB. And this is actually one of those. And it's my pleasure to introduce today's speaker, Armita Nourmohammad from University of Washington, Seattle. So Armita is an associate. How about it now? Okay, sure. Armita is an associate professor of physics and applied math at University of Washington, Seattle, and she's an affiliate faculty at Fred Hutch Cancer Research Center. Armita obtained her PhD from University of Cologne in 2012. Then she did her postdoc at Princeton. In 2017, she joined Max Planck Institute at Göttingen, and she then actually moved her research group to Seattle in 2019. She works at the interface of statistical physics and biology, and her work focuses on evolution, immunology, and protein science. She's a recipient of NSF Career Award, NIH Mira Award, and recently got APS Early Career Award. Welcome, Armita. One more? Sure.
All right. Thanks a lot for a very kind introduction. I guess everyone hears me in the back as well. All right, great. Yeah, so my group, we're a theory computation group at the University of Washington with a mix of physics, math, and computer science members. And the talk I'm gonna, the work I'm gonna present today, you'll see evidence for all of these departments and expertise in it. So in the past, I guess almost a decade now, I've been interested in immune system and how we can understand immune system from physics and evolutionary point of view. And just to give you a very, very simple overview of immune system that you probably agree with. So in the game of immune interactions, the immune cells are usually the good actors. Then pathogens like COVID will be the bad actors. And so if things don't go well, we can get sick. And as simple as that, but we still can't predict when you get sick. And so what we are interested in is trying to understand how interactions between immune system and pathogens could lead to different outcomes in hosts. The first line of defense in our immune response is called innate immunity. So this is everything that is unspecific, including your skin barrier that keeps pathogens from getting in. Then once infection sets in, your adaptive immune system starts working. And so adaptive immune system is something that is specific to vertebrates, including us humans. And it is a very unique system. So the main actors there are B cells and T cells. So you might know the term antibody, be more familiar with that. So antibody is just the receptors of B cells when they get secreted in the blood. So we have a very diverse repertoire of these cells. So each cell effectively has its own unique receptors. And we have over tens of billions of these receptors, distinct, basically, cells in our body. And the diversity of these receptors can counter the diversity of pathogens that we deal with. As a result, you can mount really specific responses to these pathogens. So just to give you how B cells and T cells differ, just very, very, again, high-level overview here. So if this is the B cell, again, the secreted detached receptors from membranes, they're called antibodies. They interact with the outside of a pathogen. They recognize outside of the pathogen. T cells, that'll be the main focus of my talk today. They see the inside of a cell, inside of the pathogen. And the way they work is if you have a cell, infected or non-infected, what it does is that a lot of the proteins in the cell get degraded over time. And so this small fraction of these proteins, right, so called peptides, small fragments, they get presented on the cell surface in these molecules called MAC molecules. And generally, when you're healthy, you're just presenting your host, your own peptides, your own proteins. But then if you get infected, you also present non-self or pathogenic peptides. And these T cells are trained when they're being produced to distinguish between self and non-self. And if they recognize a non-self molecule on the surface of a cell, they go and kill that cell, right? It's an infection, we need to get rid of it. And so if you look at this movie, what you're going to see is that you have a T cell and an infected cell here in green. And once I run it, you see the T cell start sort of attacking this infected cell and kill it. So just follow those two, and you see the sort of the merging between the two, and so elimination of the infected cell. And that happens of the order of 10 minutes or so. So once the cells come in contact with each other, it takes about 10 minutes or so to kill a cell. But immune cells also communicate with each other, so you don't need only a single cell, you need the whole army of cells. So this communication happens through molecular interactions, like signaling molecules, and it happens of the order of an hour or so. Cells differentiate into different roles, so they can become memory, that's useful for future. They can become effector, that go and really do the job during infection. So that takes the order of a few hours, and then they proliferate into larger clones so that they really go after the infection. So that proliferation can happen of the order of a few days. That's during the time you're actually starting to feel ill. And so you go from molecular scale interaction to cellular population scale interaction in the immune system. And then you can go even further. Your immune system as a sort of population, right? So human immune systems, they interact with pathogens, so influenza, COVID, and they co-evolve with each other. So we have a viral sort of escape, COVID escapes, then our immune systems, either through vaccination or reinfection, get updated. And so it goes as some sort of a Red Queen run. One is just following the other one. And that can be on the order of months to years of coevolution. And so what we are interested in our group is basically going all the way from molecular scale to population scale. And we sort of study all of these time and length scales. And what I'll talk about today is mainly at the molecular scale of these processes. To go a little bit deeper on T cells, and that's what I'll talk about today. So T cells are kind of fascinating. I mentioned they distinguish between self and non-self. So this is maybe a view of that and telling you it's actually a complex problem. Generally, a cell, even at the peak of infection, it's presenting self molecules on its MHC, right? So because there's still self protein in there, together with the non-self peptides from the infecting pathogen. So what you want is you want a response to self to be on the time scale of infinity. You do not want to mount a response to self, but you want to have a response that is really quick when you detect a non-self molecule of the order of seconds, right? So can the cell solve this problem? So the issue here is we want to have a very sensitive response and a fast response. And sensitive being the fact that even at the peak of infection, there's far more self molecules, orders of magnitude larger number of self molecules presented on the cell than non-self molecules, right? So you basically have millions of self compared to a small number of non-self molecules. So you really want to be sensitive to non-self. And the other issue is if you look at the dissociation time of self and non-self, the difference is not huge, right? So the characteristic time scale is falling of a receptor from a self and non-self is not huge, of the order of a few seconds. So if you do equilibrium calculation of binding, and so whether you can recognize cells based on just equilibrium dissociation, you can easily see that given all the numbers we have, equilibrium does not suffice this process. So if you want to actually achieve fast, sensitive, and a specific response, you need to spend some energy. It needs to be out of equilibrium, and there are actually kinetic proofreading mechanisms involved in this process to discriminate between self and non-self. So kinetic proofreading is an out of equilibrium process that you, through feedback, you try to basically be more precise in the process. And usually it comes at the cost of speed, but you speed it up by adding some interesting feedback to this, so it becomes adaptive kinetic proofreading. So this has been the work that physicists have been doing in the past many years, trying to really get at non-equilibrium processes relevant for recognition. Now, the problem that we are facing is, well, this is so for specific systems, we can characterize these dissociation times. We can go in the lab and measure a lot of these rates, and that's great. But generally, for many new pathogens, these things are completely unknown. So the landscape of immune reactions is completely unknown, and that's something we are interested in. So I already mentioned that one of the features of immune system, adaptive immune system, is the specificity, sensitivity, and its speed. The other things that I want to highlight here before getting into what I'm going to talk about in detail is that immune system also develops during our lifetime, right? So it is, as we get infected, you update your immune receptors. That's why when you get vaccinated or infected, now you're immune to the new pathogen, right? So it's a thing that is organized during your lifetime. So basically, you can recognize pathogens that did not exist when you were born. So that's kind of trivial after all this explanation, but it's still a fascinating fact. And it keeps a memory of past infection to act more readily for future encounters. So in some ways, immune system is some sort of a liquid brain. It does a lot of things that the nervous system does, but it does it in sort of more chemical reaction ways rather than the electrical reactions that we have in nervous system. So what we are interested, at least one of the things we are interested in, is how the immune system is organized to counter this diversity of pathogens that we deal with. And this notion of immune shape space was basically introduced as a way of thinking about the system in a theoretical way. It's been used quite a bit by theorists to model it. And the idea there is that if you have immune receptors in the immune shape space, when they're close to each other, that means that they recognize similar pathogens or antigens. And you have a dual space of antigenic shape space. So if the pathogens are close to each other, that means that they're going to be recognized by similar antibodies or T-cell receptors. Now, there are also supposedly some holes in our shape space, right? So those correspond to self-reactive regions that we want to selectively push out of the immune system and eliminate. So that's still debated. But generally, this is the picture. It's a nice picture, but the problem with it is that we have no clue how it's really organized at the molecular level. We don't know how many dimensions it has. We don't know even what the metric in this space is, what are the distances defined. And that's something I've been interested in, trying to sort of get at that using both co-evolutionary modeling and also looking at sequences of these receptors, because these days we can really go and get DNA sequence from all these receptors. But after a lot of work on these aspects, what we realized that at the end of the day, immune pathogen recognition is a problem of protein-protein interaction, because for most part, well, immune receptors are proteins. So here, what you're seeing here is a B-cell receptor or an antibody interacting with a protein antigen. And then here you have a T-cell receptor interacting with a small antigen. So this is this kind of curved line here, the loop, presented on the MHC molecule. So T-cell receptors always interact with protein fragments. So it's always a protein-protein interaction, but antibodies can also interact with other types of molecules. But for a large part, we are dealing with the protein-protein interaction problem. And what we were trying to figure out is, okay, can we maybe solve the harder problem of protein shape space first and go back and answer the simpler problem of immune shape space? And this might be a ridiculous way of going around this problem, but I wouldn't have been here if this didn't quite work. Before getting into details, let me acknowledge all the people who have been involved in this. The work that I'm going to present started with a graduate student in the group, Mike Cohn. At that time, we didn't know anything about proteins or machine learning. That's what I'm going to talk about. And so Mike basically pushed this project forward. And since then, after showing some promising results, there are more people who joined the protein subgroup of our group and started working on various of these problems. So a lot of work I will present today is the work of Gianmarco Visani. And fantastic collaborators at the Hutch, Dino, and also experimental colleagues at the St. Jude Hospital. Okay. So the starting point of protein paradigm is that you start from a protein sequence. So this is an amino acid sequence, bunch of letters. Then it can simultaneously fold into a protein structure, so a three-dimensional structure, and then that determines the function of the protein. There's been a lot of hype and discussion about protein folding challenge and AlphaFold. Basically, so there's been a lot of computational work, and so in 2021, AlphaFold basically had this impressive engineering effort to use machine learning to reliably fold sequences to structure, right? So that led to the last year's Nobel Prize in chemistry. There's been also a lot of work using machine learning to go from sequence to function, protein function. And that goes under the umbrella of sort of large language models for proteins. These are ChatGPT of proteins. So instead of training them on Reddit, you train them on protein sequences to get these things, right? And so they work to some extent, but they're really, they're large, as the name suggests, and they're difficult to interpret what exactly they're doing. And so in both of these problems, if you go from sequence to structure or to function, what you end up actually doing is that you need to account for a lot of long-range interactions in your problem. Because things that are far away in sequence, they can be close in the structure. And so what actually matters is through interactions, is this local interactions. And even if you are interested in local interactions, you still need to account for a lot of long-range interactions. That makes these models quite difficult to deal with because it's hard to interpret what your interaction sort of proximity actually means. So what we thought to do was, well, what if we just forget about sequence, and instead of looking at sequence, we instead go from structure to function, have a model that takes the structures as input and try to predict function. And the reason we thought that's a reasonable thing to do is that it can be more generalizable. We are learning local interactions in this case. Now, the obstacles is that we have an issue with data imbalance. So sequences are really convenient because we have so many sequences out there. So if you go to some protein data database, so UniProt is a database for protein sequences, you have billions of sequences already deposited in this database. If you go to the corresponding database for protein structures, so it's called PDB, so you have hundreds of thousands of structures, and then if you add AlphaFold structures, so you get a few million more. So this is far less sample than this one. If you're interested in specific structures, say antibodies or T-cell receptors, so that's what we are interested in. So you have about 1,000 here and about a few hundred in T-cell receptors. So these numbers are really small, even if you were to train a normal statistical inference model. And so if you want to do sort of fancy machine learning, thousands or hundreds is not a number you want to be dealing with, right? But our hope was if we learn sort of a local model of protein structure environments, and that's really looking at local regions in the protein structure as inputs to our data, prior model. So whatever we learn here, it should reflect something about interatomic interactions. So really the physics of interatomic interactions there. And as a result, it should be able to generalize the proteins that you haven't used in your training data, meaning these immune receptors we care about. And also it should be able to be used to infer function, right? So this was our hypothesis that we started with, and we were hoping that it would work, and it works quite well in some cases, as I will show. Okay, so this is how we're going to set up the problem. Let's say this is your protein structure, right? And I'm interested in local interactions. So what I will do is I will focus on a specific amino acid here. This is the orange thing. And I strip off all the atoms from this amino acid. So in a computer science lingo, you mask this thing. And then you ask the question, can you predict the identity of this masked amino acid given the atomic structure around it? So these ribbons and lines basically is some sort of a guiding way for your eyes to see the protein structure. In reality, you have basically a point cloud of atoms sitting around that area. And that's what we deal with as input to our model. And so The reason we thought this is an interesting problem, because this is basically a building block of a larger protein structure. I'm learning some effective potential for my amino acid at that position, given a very local environment. And now if I want to stack up a bunch of amino acids, I can just use these as Lego pieces and put them together. So let's see how that works. So before showing you the result, let me have a short interlude of deep learning and how we are using it for these problems. So a general sort of neural network or any inference algorithm for that sake is that you have some input, you have some output, and you want to relate input to output, right? So you want to learn a function that does that. And so the neural networks basically approximate this function using sort of stack of linear and nonlinear operations. So the linear learnable linearities, so these are these weight matrices, right? And when you're training a network, you're learning the elements of these matrices. And then the nonlinearities, which are called nonlinear activations, they're usually set up front as some sort of a nonlinear function, ReLU, sigmoid, or however you want to do it. And it's known that these structures are universal approximators to any function. So you can learn anything if you spend enough computing power to do that. And when you're training a network, so if this is the space of all possible maps, all possible f of x's, let's say you have a true map here, and maybe you have some degenerate solutions, and you're searching this space to find the true map, ideally. And what we were interested in is we were interested in constraining a search to maps that respect the physics of the problem. And I'll be a bit more specific as we go forward what I mean by physics-respective maps. So how to respect physics, right? That's a hard question in general, but for neural network, it's particularly difficult. But one thing that maybe we all agree on in the physics department is that when you're writing a physical law, you think about the symmetries that are involved in your problem, right? So let's start with the symmetries. And so that goes back to the principle of covariance by Albert Einstein, right? So that the laws of nature should not depend on your reference frame. If I am standing still with respect to a charge, I'm just measuring electric field. If I'm moving with respect to the charge, I'm measuring both electric field and magnetic field. But the universe has not changed. What we have in our theory of electromagnetism is that we have equivariance between electricity and magnetism, and we can go from one to another, right? Same goes for acceleration and gravity. So we want to have laws in our neural networks that basically allow us, as we are distorting our data, that allow us to go from one formulation to another in a self-consistent way, right? I'll be a bit more specific about it. Okay, so maybe we can't specifically learn the physics-respective map, but maybe we can try to learn symmetry-respecting map, and hopefully, the physics ones would be a subset of the symmetry-respective maps, and hopefully the separation is not too large, and so we are still doing something sensible here. All right, so let me introduce you some of the jargon that I will use. The first thing I think everyone agrees, we know what invariance means, right? So if you have, let's say, a water molecule here, and you measure its atomic mass, this quantity is invariant on the rotation of your molecule, right? If I rotate my molecule with some operator dr1, I measure the atomic mass with some function f, right? F of x being the input, is equal to f of dr1 applied on x, right? That's invariant, so that means that there is an identity operator taking this one to this one, right? Now, there's a more generalized formulation of that called equivariance. And that's when, let's say I'm measuring dipole moment in this case. And so if I rotate, the dipole moment should also rotate. And so now I need a new operator that acts in the space of dipole moments that does this rotation for me. It's called that dr2. And as long as this dr2 is a well-defined operator, I can write it down as a linear operator. We call this an equivariance. So basically, dipole moment is equivariant on the rotation of my water molecule in this case. So what we want to have is we want to have a model that I will give the model the protein structure, so atomic point cloud. I want to first encode this structure in a rotationally equivariant form. Then I want to learn a neural network model that is also respecting the rotation. So I want it also to be rotationally equivariant. And then I will use the model to predict function. So you won't be surprised to see that there will be things like spherical harmonics popping up to actually do these types of equivariance. And these ideas of sort of injecting symmetry into neural networks started some years ago, around 2016, on the group equivariant neural networks. So it was started off as a very theoretical approach to machine learning, and now has grown and shown actually quite a bit of promise in protein science, in material science, and various fields. Now these days, it's called geometric deep learning. Some of you may have heard about that. But yeah, so we started it basically reading through those mathematical papers and thinking about how can we use these things for our problem, and that's how we started in this field as well. All right. So let's see how we can actually assure that we have equivariance in our network. So the first thing that we think about when we have symmetries is, okay, maybe Fourier transform is the way to go, right? So the usual sort of domains that we think about, right? So it's when we are thinking about translational symmetry. So if we have signal in some spatial domain, right? So you can do a Fourier transform of that. You expand it in the frequency domain, like this, right? And these are your Fourier coefficients. And so Fourier transform, this sort of the usual Fourier transform we do, is equivariant under translation. So that means if I move my signal by some, some distance a, right, I get a Fourier coefficients to be basically transformed by a factor that is e to the IKA. So e to the IKA is that DR2 that we had in our equivariance slide, right? So that's as a result, right? So through translation, the Fourier transform is, this type of Fourier transform is equivariant under translation. Now we are interested in rotation, right? Because what the goal is, if I rotate my whole neighborhood, the energies should not change, right? I want to have a rotationally equivariant network. So the problem should have rotational symmetry. So if this is my point cloud and I characterize it by some density in space, the way I can do it, I can do also a Fourier transform, but in this case, I do a spherical Fourier transform. And instead of e to the minus ikx, I use spherical harmonics, right, the YLMs. And that's what it does the job for me. So that takes care of your angular degrees of freedom. So you can also have radial degree of freedom, but the radial degrees of freedom are invariant under rotation. As long as you just transform them in some well-defined way, generally you're fine. So we use some basis function for that. And then if you do this type of Fourier transform, the spherical Fourier transform, and you rotate your object, what you get is that your transformation is now, your coefficients are transformed through Wigner D matrices, right? So again, it's a well-defined operation, and that gives us the equivariance that we needed, right, to achieve the rotational equivariance. Okay. Now, the other thing I mentioned at the very beginning when I introduced neural networks that needs both linearity and nonlinearity. So this part is everything is linear, right? I rotate things to a linear operation. If I add the sigmoid functions or ReLU functions, then I break all the good equivariance things that I actually built in. So how do I add nonlinearity? So we go back again to quantum mechanics and think of, if spherical harmonics were used to characterize angular momentum, right, in your hydrogen atoms, then we can think, okay, but we have angular momentum interactions, right? That's the bilinearity. And when we want to do that, we usually use Clebsch-Gordon tensor products. So that's really what you would end up using in these networks in place of your nonlinear tensor products. And so if you do that, so you have two sort of sets of Fourier coefficients, and so then you can do a tensor product of the sort of your representations, and you still live in the space of spherical harmonics. And so everything remains equivariant in this case. So it's not a general nonlinearity, it's a bilinearity. It's between the two. But what we've seen is that it's basically sufficient to learn any reasonable model we want. Okay, so these are sort of the key ingredients of the models we're going to learn. And so let's see how we do the models, right? So this is, say, our neighborhood. We first split it in different point cloud channels. So let's say I take all the carbon atoms here, and so they're distributed in space. I take the sphere around them, oxygen, nitrogen, and so on and so forth. So these are different channels. Then I do the encoding using this spherical Fourier transform. So no neural network so far. And what we have seen is that the type of encoding we do, it's a superposition of spherical holograms, like if you were to actually make a hologram out of this thing. So we call this holographic encoding for, I guess, sounds good. Yeah. And then we have this special neural network that uses Clebsch-Gordon tensor products and all of those things, right? That takes these Fourier transform data and then lives completely in Fourier space. And at the end of that, what we end up getting is some sort of score or pseudo energies that we have associated with the 20 different possible amino acids that you can have at that position. So that's a masked residue. We don't know the identity. And if you then apply some sigmoid function, you can then interpret this as the different likelihoods, the different probabilities of these 20 amino acids at that position, right? So this would be a normalized vector. So because we are looking at local neighborhoods, right, I told you we have about 100,000 structures total in PDB, but these are 10 angstrom neighborhoods, so I can take from all proteins. And so the training data we have is about a few million neighborhoods that we trained on. And it's a simpler problem than trying to solve the whole protein all at once. All right. So how do we do on the problem that we set in? So this is what's called the confusion matrix. So these are the list of the 20 possible amino acids. So y-axis is your input. The x-axis is the predicted value. And this is average over all the sort of test data that we had. So if I was doing perfectly, I would be fully on the diagonal, right? And what I'm doing now is overall accuracy of about 70%, which happens to be a state of the art for this particular problem. But the other methods are not that much worse. But what is interesting as well, the mistakes we are making actually make sense. So if you look at how amino acids cluster when I make a mistake, things cluster by the physical-chemical properties, and they cluster by size. And what we know in biology is that you can substitute one amino acid to another as long as the properties and the shapes are not that different. It's actually fine, right? And this is more or less reflecting that. The interesting thing that we get a side note is that the training of this network is towards a magnitude faster computationally than training of, if you were to treat these sort of point clouds as 3D images and run a normal neural network on it. And the reason for that is that by constraining the symmetry in the problem, you're not searching the whole model space, but you're searching this symmetry respecting set of models. So it's much more efficient in searching the space. And so you can do a lot more with these networks. All right. So the first thing I mentioned is that, can we learn a local model? Yes, we did. Now let's see if I can do the other things. So the first thing is this model reflecting something about physics? So the easiest way of thinking about it is that, okay, if you're looking at a crystal that is frozen, right, it's probably at some sort of minimum energy in some energy landscape, right? And if I distort it, that means that the energy that I get, right, the physical energy should increase. So if my network has learned some sort of effective physical potential, the true crystal structure should be at the minimum of this potential, right? And we can try to ask that. difficult to do it experimentally, but you can computationally really distort proteins. And what we did was basically we twisted the proteins. It's called shear perturbation. And what you're seeing here is just a few examples of the things. So this is a protein called protein G, and we can go around the whole protein at each site of the protein, just twist it around the backbone. And you can look at the score or the energy we are reading off our network as a function of the distortion that you're getting. And what you're seeing is that in most cases, the minimum energy is at zero distortion, and you get some sort of parabola-looking landscape out of it, which, we didn't put it in the network, but it seems that the network has implicitly learned this type of physical potential. So that's the useful thing, so we can then use this potential to do other things later. All right. The other thing we want to do is the practical question: can we infer function and generalize to other proteins? So we have done a lot of different function inferences, and I'm not going to go over all of this. I'll only talk about the stability effect prediction in proteins, and then I'll talk more about generalization to immunoreceptors. So stability prediction is an important problem, right? So you want to make a mutation in a protein, you change one amino acid to another, and you want to ask whether that destabilizes protein or not. And so this, the protein T4 lysozyme is one of the sort of model systems of stability prediction. We have so many protein structures of these T4 lysozyme with different mutations in them. So this is the Y type, the original one. And so if I make say a mutation here from an orange amino acid to yellow, I'll get a neutral mutation, so it doesn't have a destabilizing effect. If I go from purple to pink, it's a destabilizing mutation. And we have over 40 of these structures. And in each case, we know the stability effect, the change in the free energy in the protein as we make the mutations. So that's a useful sort of test to go for. So stability effect is basically called delta delta G, is the free energy of the Y type minus the free energy of the mutant. How do we get that, right? So if you look at the Y type, we can take the Y type structure and calculate the free energy of that. Our model doesn't give you really the free energy, but we have proxies for free energy. The way we do it, so this is a sort of a weird plot if you're not used to this. Each line here shows you the letter in the Y type, the position in the Y type, and the mutation out of that letter. So it goes from I at position 3 to P. So that's one mutant. This column shows the log probability of the Y type amino acid on the Y type structure based on the model that we have, right? I give the Y type structure to our neural network. I ask, what's the probability of seeing actually this particular amino acid at that position? So this is basic that log probability. Now, if you want to calculate, so we think of this as a proxy for free energy, and it's not accurate, but let's go with that. So then we can do the same thing for mutants, but when you're looking at mutants, for each row, you need to use the correct mutant structure, right? So 40 different structures. And you get the same values, right? And so then you subtract them. And so that's a delta log P. And red is bad, blue is good, right? And so then what you actually know here, so we know which is destabilizing and which ones are neutral based on experiments. And so we see that qualitatively we separate red and blue quite well. And like quantitatively, you can actually look at the predictions, delta log P, versus the true stability effect measured experimentally. And in this case, we get about 80% correlation in our measurement. So this is not random, and like if we do these things correctly, maybe you get something useful out of it. Now, it's not always that we have all these protein mutants, right? The structure of all the mutants. And when you make one mutation, usually AlphaFold or things like that, they're not really good at finding now the mutated structure. So this is small perturbations, they're not still very well done. So we thought, okay, what if we only use the Y-type structure and you can look at the delta log P for the Y-type structure. And so this is basically done now on a much larger set of mutations, so about 3,000 mutations. And it's done by some sort of So updated version of our original holographic convolutional neural network called Hermes. So it's a Hermes model that is basically the same model structure, but a lot more modular, and you can do a lot of other things with it. Yeah, so the different axes here show different ways of measuring accuracy, and Hermes are the blue ones, and we're comparing to competitive models called protein MPNN. And overall, across all these over 100 proteins, we get something around 50% or so correlation with the true values of delta G. You can fine-tune these models to now try to learn stability effect, and you can actually boost your correlations to much higher values in that case and be more precise. And so this is actually, for structure-based models, this is now state-of-the-art for stability prediction. Okay, so again, we've done other function predictions. I'm not going to go into that. But what I'm going to get into is trying to look at T-cell interactions with antigens presented on MHC molecules. And the questions we were asking is, we have shown in other work that we can actually predict binding affinity of complexes, and this is really a binding problem, right? What I'm going to tell you about is trying to predict affinity of the interactions and try to design peptides for these interactions. We have done other types of designs in this problem. I'm not going to talk about that. The challenges of these problems is that these interactions are really of low affinity. And so micromolar, so a typical antibody, okay, we're good? Okay. The typical antibody is about nanomolar, so T-cell receptor is a micromolar, which is now 3 orders of magnitude weaker affinity than what you get as in antibodies. The interactions are degenerate. It means that there are many T-cell receptors against the same antigen peptide, and there are many peptides against the same T-cell receptor. So that's a problem. Having a low affinity also resulted in scarcity of structural data because it's hard to stabilize these things to do X-ray crystallography or cryo-EM. So these two go hand in hand. So basically what we decided is that we should not train on T-cell receptors because we don't have that data, but we take our model of protein universe and try to basically test on these things and see what we get. All right. So why is this an important problem? So this is a picture we started with. So we had the self and non-self. But sometimes the self proteins actually can acquire cancer mutations, right? And what we hope is that we should get to push our immune system to recognize those cancer mutants and kill the cancer cells, right? So that would be an ideal situation. And so the cancer mutant, right, the peptides that contain the mutations also presented on the cell surface. Now, the question we have is that how well our T-cell repertoire, right, T-cell receptors can recognize these cancer mutants, cancer peptides, what's the limits of recognition. And now, if we design a T-cell receptor that is reactive, can we design a T-cell receptor that is reactive to a specific antigen, to a specific cancer peptide? And if so, does that receptor also bind to other self-peptides? And that's the main problem with these type of receptors, because if this is just one mutation away from the original self, due to cross-reactivity, once you bind to this, you bind also to self, and that means it causes a strong autoimmune reaction. So some of the T-cell therapies that we have, and I'm going to show one of those later, like they actually cause cardiac arrest when you introduce that therapy to the patient, because it was cross-reactive to some heart protein that was expressed in the patient's heart. So do the design, so can we avoid autoimmunity? So what's the specificity of this interaction, in other words? And so the last question is equivalent to what's the space of peptides that a given T-cell receptor can recognize. And so that goes to the problem of peptide design, and that's what I will show you. So it turns out, I'm not talking about it, it turns out that it's really easy to actually design T-cell receptors because they have low affinity. You can easily boost up their affinity by making them highly charged. They bind to stuff. But they're very sticky. They bind to everything. And so problem with specificity is actually a much harder problem. So the first step is measuring the affinities. Again, very limited data that's out there that we can use to actually test our predictions. So this is two systems, and you have one MHC, two different T-cell receptors, very different, and a sets of peptide variants in each case. So one is called tax family, and the other one is NYESO, and don't ask me why they're called the way they're called. And so they're also measured the binding affinity of these peptides to these T-cell receptors for 16 variants in each case. Again, very small numbers. But so this is a case of NYESO, all the data points, so we get about 72% correlation. And what we see down here is also specific sort of values of binding affinity versus our predictions for different mutants. This is the structure. And for tax, we have a lower affinity. So again, small numbers, it's really hard to judge if this is doing a good job or not, but it's a good start. The other thing we can do is that instead of affinity, which really is a difficult thing to measure, you can measure the activity of T cells, right? So you can pulse a T cell with a peptide and then go and see how active this T cell is, how much signaling molecule is producing, and that's a much easier experiment to do. And so this is data we got from activity measurements of many, many different peptides in the context of pancreatic cancer immunotherapy. And so the way they did is that they take one peptide, you have a wild type, you have the mutant, and you change the concentration of the peptide, you read off the activity of the T cells that you're testing against, and so you can measure this sort of half activation type, or EC50, that would correspond to some measure of activity of that peptide, right? And so the difference between mutant and wild type, if the mutant needs more or less peptide, it shows you it's more, have higher or lower affinity. So this is sort of a panels of these peptides. In each case, we have all the single point mutations starting from a wild type. The top, these are some viral peptides from CMV virus against different types of TCRs. And then below is melanoma peptides. So these are tumor peptides, again, mutants against a bunch of different TCRs. And so except for this one, everything else is alpha-fold folded structure because we didn't have the structure in those cases. And what we see is that there's a range of sort of correlations. You go from 70% down to effectively 0 in this case. But yeah, so roughly you're living in the area of like 40 to 50% correlation. The case that we have almost 0 correlation is the case that AlphaFold actually is doing a really bad job in folding. AlphaFold has this really cool feature that it gives you how confident it is about the job it's doing. And so this is, for this system, is a bad error. Higher is worse. And so in that case, we are also, when you're giving garbage, you're getting garbage out. And so the other system that we did is like similar setup, but using antibodies, and then you get also around 50% correlation with the experiments. So these affinity measurements are encouraging. Again, nothing is 100%, but if you think about exploring a space of million sequences down to 1,000 sequences that you can go test in the lab, that's already a win, right? And so what we thought was, okay, if we take our predictions a bit seriously, can we use that to design new peptides? Can we sample from this energy landscape for peptides? And this is specific structure. And so the idea here is, okay, if I'm trying to design this peptide that's 9 to 11 amino acid long, I can fix the T-cell receptor, I can fix the MHC molecule, there's the backbone, and then I sample side chain, which basically is the identity of the amino acids using our model. Then I relax these structures, measure energies, right, using Monte Carlo sampling, and then I iterate until some sort of convergence. And maybe I get a peptide, I can filter it with some other tools like AlphaFold or related tools, and then I can go ask my experimentalist friend to actually go and test our peptide. So that's what exactly we did, right? So we did it for a whole bunch of systems. I'm going to show you only one, to sort of test this procedure. So this is a system I already mentioned. It in the original therapy. So this the T-cell receptor is an engineered T-cell receptor against a peptide that is common commonly seen in certain cancers. And they use it for therapy, and it actually led to the death of 2 individuals because of the cardiac arrest through the cross-reactivity. But what we wanted to do was, okay, can we make the interaction even stronger? And can we design peptides that are good for these T-cell receptors? And so this is the set of design peptides. So we had 93 designed peptides in this system. What you're seeing here, the 2 colors are the 2 versions of our model, so you can ignore. These guys are the wild types. This is the original MAGE peptide. This is the one that led to cardiac arrest, so it's a higher affinity. So the y-axis is your T-cell activity experimentally measured. Higher is more active. This is a control when you don't put a peptide. The dotted line shows the activity threshold that we have experimentally. And as we can see, the overall accuracy is about 20%. If you limit it to things below 6 amino acids, which over many experiments, we've seen that this is a good range, you get up to a 30% for this particular system. And what does that mean is that if I want to go all the way to 5 amino acids, I'm actually looking at the space of 10 to the 8 possible peptides. So when I'm saying, okay, I have 30% or 20% accuracy, that's already quite a lot, right? So because I can go in the lab and test 1,000 peptides with our designs, and 200 of them should work. And so, but if you just randomly choose one sequence from 10 to the 8, basically you can be sure that it's not going to work, right? So in the other 2 systems we tested, that's also one of them was cancer and the other one is a viral infection. We get about 40 to 50% design accuracy. We were also interested looking at these guys that we end up actually having larger activity compared to the original white type. So we wanted to see what's happening and what have we done there, right? So to improve it even further. And so if you look at the protein structure, so this is the original MAGE. This is the one that causes cardiac arrest, and this is the one that we designed, one of the best one that we designed. And what we saw is actually there is this mutation at position 8, right? The prevalence of the amino acid E. So, okay, so let me walk you how to read this plot. So these are the different positions of the peptides. You can think of the y-axis as frequency, but it's in bits, so it's log frequency or p log frequency, right? That's why it's sort of entropy. So the larger the amino acid is, the more prevalent it is in the sets of things that are working. And what we're seeing is that compared to improved design, if you compare improved design to all the positive designs that we have this amino acid at position 8 to be very, very prevalent. And then we can go in the structure and see what it does, is that it's forming polar bond to some region of the T-cell receptor. And what we compare is that this is a very strong polar bond. We form actually already a slightly weaker version of that when you go to the cardiac arrest peptide. And that polar bond didn't exist in the original peptide. So that's like, you can really go back and identify the mechanism out of this highly statistical, a model that was not even aware of the T-cell receptors. So that's encouraging. A few minutes, or okay. So that was sort of practical thing, and we were designing peptides to do useful things with them. But then going back is like, okay, now I have this powerful tool. It's not perfect, but I can use it as a proxy to measure, to say something statistical about the space of possible peptides. And let's try to do that. So this is a structure of an MHC molecule with a peptide. So the way it says, MHC is presenting the peptide. And if you look at the structure, you see that in the interaction, the MHC has basically, the interactions you see is at the anchor position, at the 2 ends, you form some polar bonds. And we have a whole bunch of MHC molecules that we can go, there's a lot more, but these are the ones we have the structures for. And you can measure basically based on the distribution of peptides that each MHC molecule can bind to, you can measure the entropy of that distribution. And so this is more or less what you're seeing here. should look at it on the sort of horizontal way. So you get something around 29, 28 bits of information or entropy, right, for the peptides that bind to a specific MAC on average. And so if you then translate that bits to a typical number as 2 to the power of entropy, you get about 10 to the 9. So each MAC can present about 10 to the 9 different peptides in humans, right? So this is an MAC, but what we need is T-cell receptor as well. And the question we are asking here is that adding a T-cell receptor to the pool, how much is going to restrict the number of peptides that we can recognize? So this is the MAC peptide and now a binding T-cell receptor. And if we look at the interactions, typically what you end up seeing is that there are all these polar bonds that are formed between the T-cell receptor and the peptide that restricts the identity of the peptide. And so we can do the same game here. And here is a distribution of, each point is one protein structure, basically, on these plots. And the number is the number of structures that had that MAC, but many different TCRs. So we get something much smaller, so about 10 bits or so of entropy, which translates to about 1,000 different peptides. So a TCR MAC specificity ends up being about 1,000. So once you go from 10 to 9 to 1,000. You can do the same exercise in mice. We have much less data in mice, but you can do the same thing. And so you get roughly the same numbers in mice as well. So these kind of peptide design approaches, they can be practical, but they can also help you to answer more sort of fundamental questions about these shape space and the specificity landscape of T-cell receptors. So with this, let me just summarize what I told you about. So we have this generalizable physics-guided structure-based model, which we think is useful for modeling proteins. It can be used to predict T-cell receptor peptide MAC interactions. Again, a useful thing for peptide design. So that's good for vaccination, so cancer vaccines, viral vaccines, and other things, and also useful for T-cell receptor design for immunotherapy. As I said, yeah, so we can design peptides by drawing from this landscape, and we can use that to then characterize our specificity landscape. With this, I'll thank all the people who were involved. So I already mentioned most of the work was done by Gianmarco Visoni, all the things I presented today. The beginning of the work was by Mike Poon, who is not in this picture, and many others who have contributed to the work. Thank you for listening. Happy to answer questions.
We have time for questions.
Can you speak a bit louder, please?
Oh, yeah. For the generation of your point cloud when you're training your model, do you account for like B factor or temperature factor?
So not B factor and temperature, but we account for charge and surface accessible area, so SASA. Yeah, so B factors are pretty noisy, so it's not so great. We tried it, we didn't get any good. And then temperature, we don't have really good access to that, but in principle, I think for stability measurements, for example, pH is an important factor, temperature is an important factor for stability. Yeah.
So AlphaFold 3 basically got rid of their equivariant quantities and replaced it with data augmentation.
Yeah.
I'm curious if you guys try to do it with innovation to test that and how that works.
Yeah, so you can go, we don't even need to do the ablation because basically people have done this exact problem with data augmentation. You get the same result, it's just much slower and you need a lot more compute. And it's not necessarily learning the physics that we want. So that's the only difference that I can highlight. But again, with AlphaFold 3, people have been looking at equivariance and how it scales with training, and you can show that, okay, if you have infinite compute, you can do it, right? But the scaling is still in favor of equivariant models. So that's still there, right? If you don't have DeepMind compute money, then I think you should maybe think a bit more carefully about the structure of your model.
I have a question. Yeah. So you observe correlation with JP and activity.
Yeah.
But
So that's the T cell activity. So I guess so it's not exactly between KD, it's our prediction and activity, right? So our prediction and KD have generally higher correlations in other systems. For the systems we had activity with, we had a slightly lower correlation, right? About 50%. Activity does not only involve binding, but involves many other things, right? Co-receptor signaling and all those things, which are not in our model at all. So I think that's a really interesting question. We're trying to get at that, but again, we need a lot of data that you can separate, signaling from binding exactly for these systems to actually answer that question. I think there's a limit on how much you can predict, and I don't know if the limit is 50% or higher. So.
You mentioned that the binding energy between these T cell receptors and MHC is quite low.
Yeah.
So for the T cells, like the killer cell, does it need to bind lots of T cells with lots of receptors?
So you form a co-receptor. You bind with sort of next door co-receptor. So you form actually groups of receptors on the surface.
It's very cooperative.
Yeah.
And can you actually detect if it's really just one molecule or a synapse in a protein? Would you be able to detect that with a T cell with a properly designed receptor that
So you mean
mutation?
Yeah, you can do that. That you can do. You can easily know which peptide you're binding to. Well, not easily, but you can know which peptide you're binding.
Thanks for the great talk. You incorporated symmetry, local geometric symmetry, spatial symmetry to predict the function. Have you thought about incorporating time and sort of the information to?
Yep. So, sorry. So you can try to train models on molecular dynamics data, and people do that. We haven't yet. In that case, the system is not equilibrium, so it's not time symmetric in some sense. You always have a time directionality there. But yeah, you can have a conservation of energy kind of criteria to get to time symmetry. So I think that would be interesting. Yeah. You can do it for toy models that are not proteins, and you know exactly, you have time symmetric system, right, in equilibrium. And that would be fun. Yeah.
This might be a technical question, but how are your weights adjusted?
Yeah, so it's basically neural network backpropagation, right? So neural networks do this automatic differentiation, which basically, your Newton's chain rules, right? So for differentiation, and so you have a loss function that you're trying to minimize. That's the similarity to the input, and so you basically perturb it, and you want to see if you're going down the loss or going, So and you change the weights, or you calculate the gradients of the weights, and you go along the direction of those gradients to basically go down the loss function. You update all of them, and so you actually just measure the gradient. So whatever, you go along the gradient by whatever magnitude that gradient is, right? Yeah, so it's like if you have a very large gradient, then you go more down, right? Yeah, but yeah.
Do you have like one more burning question before we close?
Right.
All right, I think that's it. Thank you so much.